Good morning, everybody. We would like to start the second session of today. And I have the great pleasure to invite Dr. Olivier Rabin, the WADA Science Director, to develop for us the relationships between WADA and the pharmaceutical industry.
And from there?
Yes, please.
Okay, thank you, Costas. Dear colleagues and friends, ladies and gentlemen, I'm very pleased to be with you today, and I would like to start by thanking the organizers of this meeting, and in particular, Dr. Mohamed Al-Zairafi, for giving me the opportunity to be with you today. It's always a pleasure to be here in Doha, in Qatar, and having the support of colleagues and friends, not only in science, but globally speaking, in the fight against doping in sport. I was asked today to give you a little bit of information about a project that I've been following at WADA for more than 10 years now, which relates to the connections with the pharmaceutical industry. And certainly, one of the first questions that we might raise is why do we need to collaborate with the industry? And I would say there is an observation and certainly a statement as well, is that, first of all, the observation is that the vast majority of doping in sport is coming from the diversion of legal drugs by the athletes, by far. When we talk about EPO, when we talk about human growth hormone, these products have a legitimate therapeutic use, and they are diverted by the athletes to enhance their performance. And also it came with a WADA mandate, which I think for the first time started to look at the future and say, you know, what is going to be doping in the future, in the 5 and 10 years to come? And when you combine the two, of course, you start to look at the new drugs that are coming in the pipeline of the pharmaceutical industry. And so I started this, and probably one of the first elements that we need to be aware of for those of you who are not necessarily working on a day-by-day basis on drugs, is that, and this is a little bit artificial, but I think it helps to understand the global concept of the different drugs we are facing in the fight against doping in sport. I was talking about the legitimate medicines, so medications, and usually these drugs, you've got a wealth of information on these products. Not only do we know the active principles, but we know a lot about the pharmacokinetic of the drugs, the properties of toxicity, pharmacology, and we have access to the reference materials, and sometimes to the detection methods, which is very helpful to transfer to the anti-doping laboratories. Another class are the drugs that are not known yet, and some of them may never be known, may never reach the market, and they are the drugs under preclinical and clinical development by the pharmaceutical industry. And what is very important to understand in this situation is that the industry does not need to communicate about these drugs. They have the entire responsibility up to the end of the clinical development. So usually very little information available, high confidentiality, because you imagine the amount of money that is at stake and the need to keep this information confidential. And as I said, it's the responsibility of the company. Not even the drug enforcement agencies can force the companies to communicate on drugs in development. And finally, the illegal drugs. I mean, you heard David Holman this morning talking about the underworld. This is a reality. Designer drugs, street drugs. To give you an illustration, in 2011, there were about 40 new designer stimulants that were identified. In 2012, there were 72 or 76. So this is a growing market, and we are facing these products, just to name a category of product that we are concerned about in the fight against doping in sport. So this is the sad reality, but something we have to face when we are talking about drugs in sport. Back in 2003, when I joined WADA, and WADA was the embryo of what it is today, fortunately, I would say, and anti-doping in the world was not even what it is today, we started to think about what are the drugs that are going to be the major threat in the coming years. And because before I joined WADA, I was in the pharmaceutical industry, I came to WADA with the tools how to identify these drugs and how to have access to these drugs and how we could use this information for the benefit of the fight against doping in sport. So this is something that we started to identify, and we started to think about how to approach the pharmaceutical companies. And needless to say, and think back in 2002, 2003, as I was saying, WADA is not what it is today. And literally, I had to contact these companies and spell WADA, what we are doing, and what we wanted to achieve with their collaboration. A lot of negotiations to allow them to share information with us. And I would say mainly a lot of perseverance and patience was needed to really convince them to work with us, because as I said, not even drug agencies can force the pharmaceutical industry to share information. Can you believe WADA in our position to encourage them to share information was even a greater challenge, of course. Quickly, we had some very good successes. And I give you here 3 examples because they are very representative of some of the issues we faced. First of all, with Roche, big pharmaceutical company developing a drug for anemia. This drug happened to be of high interest and concern to us in anti-doping, and this drug, Sierra or Mircera, was certainly something of a real issue that we identified in our net of drugs of concern, of high concern, I should say, certainly on the very top of the list. Another example is similar category of drugs, and I selected those examples on purpose to remain within the same blood manipulation, EPO-like drugs. The concept developed by Affymax, a very small biotech company located in California, the very opposite of Roche. And we convinced them to work with us. We really approached them on a very personal and very friendly basis. We talked to the very top people in the company and convinced them of the issue. And it's a radically different approach when you talk to a big pharma, well-established, well-known all over the world, a global company, to a very small company. But still, we succeeded with the 2 kind of partnerships and convinced them to work with us in anti-doping. Another example, Shire, that had developed at the time the EPO delta, which was of high concern because it was so similar, or supposed to be so similar to the human endogenous EPO. So all these illustrated some of the issues we faced very early on in our project when we thought about discussing with the pharmaceutical companies. And again, these are only a few examples. But after the examples, and first, I would say examples of success, the question was about how to become more global in this approach. How can we convince the industry to be a global partner? Not only in the collaboration agreements, how to go away from very focused discussion on one drug or a class of drugs, how to expand the partnerships, not only to have access to more information, but force the pharmaceutical industry, or encourage, I would say, the pharmaceutical industry to think in a broad sense about what we are doing. How to also inform and educate. It's the idea, in fact, the concept was WADA is small, in a sense, in particular in those years. The pharmaceutical industry is big. Hundreds of thousands of collaborators around the world. How can we empower the industry to help us in what we are trying to achieve? And also maintain the interest. There is a high turnover in the industry and people lose interest. They are like any of us. They've got different priorities, and priorities over priorities, they tend to lose the interest. So we need to maintain this interest. And one of the first steps was certainly to not only adjust to each company, because from the outside, the organizations seem to be more or less the same. Well, from the inside, it's radically different. They have, even if they use the same global language, you've got some internal languages for each company and internal way of thinking, different cultures that you need to approach and understand so that you can adjust and adapt to the organizations. Just to illustrate this, when I discuss with the farmers, I can discuss with the top of the R&D, so research and development, because they are the ones in charge with the development of drugs, or I can talk to the people, well, the heads of regulatory affairs, because after all, safety is also part of the regulatory dossier when a drug is developed. And I can also talk to people who are in charge with safety. So it varies a lot, and within the company you need to identify the good relays and the good champions who are going to be the people who are going to understand what you're saying and who are going to carry this message from within the company. And this is very important to identify those people and identify the decision makers whom you can try to influence so that in the end these people are on board and they are going to make the step of collaborating with WADA and with the anti-doping community in general. Flexibility in the agreements. It's clear that if you arrive with your ready-made template of your agreement, it's not going to work. Again, because companies have different cultures, have different perspectives, relate sometimes differently when they have a drug of high concern or a class of drugs or even a franchise of concern to anti-doping. People, in particular companies in the blood aspects anemic franchise are highly easy, I would say, or very easy to inform, and they relate very quickly to you. When you talk to companies who are not in those areas, for example, oncology, they don't necessarily relate very easily to anti-doping. So this is something to keep in mind. And of course, the long-term approach. It takes years to approach companies, to convince them to work with us. I can tell you there is a biotech in the US. I've been approaching them for 7 years now, and we just signed a confidentiality agreement about 3 months ago. So that's sometimes how long it takes to convince them to work with us. The kind of agreements, they are different, again, to be flexible but preserve the interests of anti-doping. We've got different form of relationships with the companies. I would say the ultimate example is the open global agreement. In other words, a company saying, we want to sign an agreement with WADA and we want to be able to freely share information with WADA on any of our drugs in development. And this is the example that's been made public with GSK, for example, or Roche. They can approach us anytime because we've got an open confidentiality agreement, and they can share with us any information or any concern they might have on the new drug. So this is something which is very productive because, again, it allows the flow of information between the company and us at WADA. There are sometimes agreements that are limited to a substance or a class of substances. This is typical of some companies that have an interest of a drug or bought a drug, acquired a drug, or a biotech company for which there is a very specific franchise or a couple of drugs that they develop, and they don't want to expand beyond this. And that's very legitimate. We don't have to force them. We simply discuss on a confidentiality basis on these very specific drugs. And we've got several companies that we approached, and they do not necessarily have a drug or a class of drugs of concern, but they are very open to collaborate with us. And they do not necessarily need a formal agreement in place, but they are happy to share with us some information or say, we'll be ready to communicate with WADA when we have a drug or some drugs that are of concern to us. And we've got several companies in that situation. Now, just to illustrate a few of those examples, and they've been made public, so there are some big pharmas, and we are currently negotiating at WADA with other big pharmas. The idea would be to have the top 10 pharmaceutical companies in the world collaborating with us, and that's the embryo, and then it will certainly spread around, but we've got discussions, agreements with other companies as well. What really matters to us is really the portfolio, and again, assessing the risk and identifying the drugs before they become a reality as a threat for the fight against doping in sport. The reality being, whether we like it or not, that today you've got some drugs in development that are already copied and made available on the internet. The agreement I was referring to a little earlier with a company we've been discussing for 7 years now, their leading product is now available on the internet, and we had access to the product on the internet before we had access from the original compound from the pharmaceutical industry. So that's a reality we certainly have to take into account in our approach. It's not only about the companies, it's also about the associations representing the companies. It's important that we can link I would say almost on a personal basis with some farmers, but it's also very important that the associations of farmers do understand what we are doing, and we can, in fact, relate to them when we've got some issues on a particular drug, I can easily contact them and say, who is your contact within this company? Because I need to approach this company. I need to have a reliable person in this company to whom I can speak and explain what we're doing. It's always a bilateral relationship at some point. It cannot be global, but we need the global coverage to help us in what we're doing. So we, very early on in 2008, signed an agreement with the French Association of Farmers, LEM. Then soon after with the IFPMA, as David Holman mentioned earlier, which was a big step because the IFPMA represents the big pharmaceutical companies in the world. So having their support was very important. And very recently, our colleagues in Japan also signed a similar agreement. And I'm currently discussing with the European authorities to really leverage their support and facilitate the communication with the individual pharmaceutical companies. It's not only about the agencies and about the farmers and the associations representing the farmers. It's also about the drug agencies. When we thought about approaching how we could develop an efficient network of people interested in this issue of drugs and abuse of drugs, it's clear that there is a role to be played by the drug agencies. It would be, I think, an illusion to believe that every single pharma developing a drug is going to approach WADA at some point, or any anti-doping authority. Some of them will fly under the radar, and they will come to the time of registration of a drug, with their dossier, and we will not have seen this drug coming. I know usually you've got clinical development, and you can see, but I can assure you that there are some small developments for some very niche products that are much more difficult to detect than others. And it's important that we've got the drug agencies on board, because these people were the ones who, in a sense, give their approvals for the drugs, do have knowledge, of course, in what we're doing, not specifically about the anti-doping aspects, but the potential effect on the physiology, the toxicity profile of a drug, is something which is their bread and butter, and it's very easy to communicate with them. So over the past few years, I approached the EMEA in Europe, the FDA in the US, PMDA in Japan, and as well as other international and national agencies to convince them that there is a link to be made with us in our community of people fighting against doping in sport, and we can exchange very useful information, and it's very appropriate to do this as part of our mandate. Just to complete my presentation, just to leave with you a few priority areas, which are areas we are particularly concerned about in terms of drug development. Of course, oxygen carriers. All of you know the development of EPO, EPO-like substances, and today you've got drugs, and I mentioned peginezatide from Affimax. That is a drug that has nothing to do with a sequence of peptides of the original EPO. So you've got drugs that are absolutely not related to the original product, still having absolutely the same effect, and we move today to the HIF stabilizers, HIF activators, and the likes. Muscle, of course, that will remain, we heard about anabolic steroids, but there are many drugs today that are on myostatin inhibitors, for example, but many others. Also, we can modify the composition of the muscle fibers and also the properties of the muscle. Energy metabolism, we can connect this to the muscle, but it's certainly a vast area of improvement, and those of you who are in sport physiology do know that optimization of metabolism is certainly something that is of high interest. And we've got drugs today, I mean, ICAR and many others that are in development that are going to act on that sphere of energy metabolism. And finally, it's a kind of global thing, but hormones and peptides are certainly an element of concern to us because we've seen quite a few of these products being developed, not always with very well-known properties, but certainly being extremely quickly copied and made available. David Holman referred to MGF. There are many of these hormones and peptides that are now available on the internet. some of them very poor quality, some of them better quality, some of them being in development by the pharma companies, but also being already copied and being made available on the internet. So this is the reality we are fighting today, and this is something that we have taken on board at WADA to try to have access to these products, certainly the original products, but also the copies that are also, sold on the internet, to have a much better view on what is being sold out there to the athletes, and of course develop our prevention side and certainly detection side to be as deterrent as we can be for the abuse of these products, many of them being of extremely poor quality, I can assure you. So I just wanted to leave with you this with this kind of feedback, I would say, on the past 10 years, and also a little bit of perspective, because I think very strongly that each of us, when we've got connections with the drug agencies or with the pharmaceutical companies, do have a duty to inform them about what we are doing and see them as potential partners. Because when they develop the drugs of tomorrow, this is what is going to be available to the athletes, either directly, as we know, and we've seen a few cases for some EPOs in the past, or indirectly via the internet. So this is a reality we're facing, and again, I think each of us have a duty to contribute to this development of relationship with the farmers and access to the drugs in development. So thank you for your attention.
Thank you very much, Olivier, for all this very important information that WADA and yourself providing us. And I would like to invite Mrs. Alanur Al-Kahtani for our young promising student and colleague in ADLQ, please.
Thank you, Dr. Kostas. It's an honor to stand here and present my presentation, which is anti-doping and beyond. Anti-doping is not just a doping analysis lab. There's much, much more being done within our great constructed walls. I'll start with talking about the Qatar national vision, because that's what started everything. And it states, in order to further support human development, Qatar will aspire to be an active center in the field of scientific research and intellectual activity. And on that basis, Dr. Mohammed Al-Sayrafi built his vision. And on that, we also had a mission. The vision of anti-doping was to be a leading anti-doping and research facility with integrity, ethics, and excellence by 2018. And to reach our vision, our mission states, is to run the organization on strategic goals that will facilitate our vision. And our mission has 5 different pillars to it. One is to be a first-class anti-doping a lab contributing to Qatar's sports infrastructure, and to promoting a healthy lifestyle, and to be a leading scientific and technology research organization, to enhance the skills and capabilities of Qataris among us in training and education, and to move forward with financial sustainability. We always need the money. And how did we align our vision with the Qatar national vision? The ADLQ strives to contribute to all 4 pillars of the vision, which are the human, social, economic, and environment pillars, by being active in the field of science, scientific research, and building scientific skills and capabilities among the Qataris. To build that, we need to have integrity. We cannot build something without any integrity, whether it's in science or in constructing our building. And the Rasul, the Prophet Muhammad ﷺ said, whoever cheats us is not one of us. And this is not only in Islam, it's in all religions and in all man-made laws. Therefore, we based our work to have integrity in sports and in research as well. Dr. Mohammed Sayrafi took all of this to construct the anti-doping lab. This is a picture of our building. So the anti-doping lab is basically the core lab, which is the doping analysis lab. And as we all know, the doping analysis lab does all the doping research, whether it's through finding different tests to be more sensitive to stimulants and steroids, or through testing for sports and activities within the athletes. On that, Dr. Mohammed Al-Sayrafi had a vision, because you cannot talk about doping and not mention toxicity. So he built a second floor that had the toxicology and multipurpose lab. And on that, we cannot have doping and toxicology without research, because everything is supported by research. And we had a third floor. I guess it just opened his appetite to build the life sciences research lab. What do we do in the toxicology, multipurpose, and life sciences lab? When we talk about these two floors, we always talk about health-related issues. Health-related issues start with environmental factors, and there's a whole list of environmental factors that we can think of. First of them, of course, the doping, and that's where the doping analysis lab comes in. We don't only do doping analysis, we also do public health awareness. We don't do the test for the sake of doing the test and finding athletes who take dope materials. However, we aware the public with the issues and the causes and the consequences that come with it. Another thing, as I said, we have the toxicology multipurpose lab. We have 4 different subgroups within the toxicology lab. The omics, the analytical toxicology from different chemical compounds that are being consumed as toxics to the human being. We have the cellular toxicology, where we do the healthy aging and the healthy lifestyle. And also we have service, whether it's for the community, for the government, or the agencies. And also we do service to the research, and that's where the research comes in. And that's our third floor. The third floor does the protein engineering, the stem cell research, the cellular histology research, the sequencing, the physiology, and of course the vascular biology. You can see this is a whole circuit that does cover everything, but this is not only the only factors that come in. We also have the genetics, the proteomics, and the glycomics. This is what we are doing. We're trying to cover everything, from a small particular chemical and how it toxins the body until its effect on the whole biology of the system, from where it starts to where it ends. And of course, we have to think of the public awareness. What we're trying to do is to build integrity and public health awareness within the community. And everything works together. Nothing is separated. Everything has to be connected to other. You cannot say we run a first floor without the second. We cannot say a second without the third. We can't say a third without the first. Everything has to be entwined together. This is a very short list of the kind of projects that are public health related that are being done in the ADL. Of course, my project is the first one, and then the rest of my colleagues come in. As you can see, I can point out the first one. Mine is drug discovery and detoxification and cancer treatment. And then the second one is the shisha water, the analysis of the compounds that are shisha water. And then we have, for example, the vitamin D testing. We have the corticosteroids for service. We have a lot of other factors that can go in. We have the obesity as an effect to and how it's public health related to the issue. With that being said, I'm a scientist. I have to talk a little bit about science. I promise it's not going to be boring. This is a short. I'll give you a short description of my PhD project, just to give you a feeling of the kind of research that we're doing. Of course, I'm a PhD student in Groningen University. This is my first year, and my project is funded by Qatar National Research Funding. The title of my project is Studies on Targeted Therapy and the Detoxification of Drugs and Cancer. We all know cancer accounts for 13% of mortality rate worldwide. And there's a lot of technologies and a lot of techniques that are put in the treatment of cancer, such as chemotherapy and radiotherapy. However, none of them is targeted, and that's what I'm trying to do. I'm going to create or try to do a targeted therapy for cancer. We're looking at an enzyme, and the enzyme I'm working with is linked to an antibody that's specific to that tumor. So once we get a non-toxic drug into the body, my glucocarbidosis, my enzyme, will convert the non-toxic into a toxic just in the presence of the cancer cell. Therefore, we are lessening the effect of toxicity of the non-specificity of the cancer treatment itself. Of course, there are a lot of treatments, and this treatment is present in the hospital. I'm not saying I came up with a new one. However, what we're trying to do is to modify the present techniques and create novel ideas to create efficiency within its application. Who's behind the success of the ADL? When we think of projects and we think of everything, how does it all fall in? Of course, we have the bosses, board of trustees, and our manager, and then we have our admission area with the subgroups. And then we fall in. I call ourselves the money makers. The life sciences research, the toxicology and multipurpose lab, and of course, the doping analysis lab. And then, of course, we need money. Where does the money come in? The money comes in with the funding. We have external funding and internal funding. QNRF are our external funders, and of course, the ADL has its own budget to cover projects. However, they have to fit within the vision. And this is a picture of our grant winners of last year. Let's hope this year, inshallah, we'll have more members joining in. Of course, we don't work alone. We have collaborations. We have international and national collaborations. And the whole idea of collaboration is to create a scientific environment where we can share knowledge and share experience within other people and other regions around us. We have an MOU signed with Groningen University, UCL, Southampton, Malaysia, and inshallah soon with the Jordanian University. And we have an MOU with Qatar University. And the rest, we all have partnership within our research. And finally, I would have to say that ADL is a story of a great success in Qatar. It's been a few years, and I believe we have made our print within Doha. We have successfully managed to be a perfect mix between the doping analysis service and the community and life sciences. And through the scientific and community activities, the ADL has established a very good reputation nationally and internationally. And finally, of course, we always have to remember our logo, be pure and stay pure. And a very big thank you from the ADL family. I believe we are the silent warriors. Thank you.
We all thank you very much, Alanut, for your very nice presentation. And I would like to invite Professor Olaf Schumacher from Aspetar Hospital. Olaf Schumacher is one worldwide expert in the new tool of WADA, which is the indirect measurement of the athlete's biological passport related in blood parameters. Please.
Thanks, Kostas. Thanks, Dr. Mohammed, for inviting me. It's a great honor to be here as a humble researcher and speak to such a select group of audience. I want to thank Dr. Mohammed especially because when he invited me, we talked about the topic, and he said, you know, Dr. Olaf, you know, doping, it's all about medication, all the medicine stuff going into the athlete. Is there also another way around? Is there something that comes out of anti-doping that can actually profit medicine and the progress in medicine? And I gave it a little thought and actually came back to that topic, and it's actually quite a visionary topic. So I'm going to try in the next 15 to 20 minutes to explain you how this new tool, this athlete's biological passport that already David Haumann, Olivier, and many other people here had talked about and alluded to in their talks, can actually feed back into medicine and the progress in these areas. So first, we're going to talk a little bit about the background because many of you might not know how it actually works. The ones that know, they can go out and have a drink. You know, so I'm going to try to explain you a little bit about the biological passport, then how it relates to health, and maybe future applications and developments and future areas of research of benefit, maybe for medicine in general. Now, Lance Armstrong is present every anti-doping talk, so also in mine, you know, but Lance Armstrong is a little bit like all of us going to work every day here in Qatar, driving our cars through the streets of Doha. We're all a little bit Lance Armstrong, and you will ask why. Well, the solution is really, really simple, you know. Lance Armstrong, as David has said, never missed a test. This is what he tweeted, you know, 20 years career, 500 drug tests, never tested positive. Of course he doped. And this is what we're doing every day with our cars. We're driving through the streets of Doha and try to avoid these signals here, you know, and not to be flashed. Of course, some of us are more successful than others, and we never test positive, although basically we speed every now and then because we have to go to work in time. Now, this is a little bit the problem in anti-doping. There's kind of a lacking efficiency, and you can compare that to these traffic flashes. You know, so a lot of controls, not many positives. Of course, many more people dope. David has illustrated that very nicely with all the citations from Tyler Hamilton's book. Now, what's the solution? Again, we can look at other examples from normal life. This is the tutor system. This is a system that is installed on Italian highways. Now, you will see two gates, and on the gates there's cameras that take the number plate of the car and measure the time between the gates, and then will calculate the average speed at which the car went. So that's all automatized. So if the car goes too fast, there you get the fine. So the system is actually quite efficient, has reduced the death rate on the roads where it's installed by 50% and the injury rate by 20%. So they are not measuring here the speed directly with the camera, but somehow indirectly. And this is also a little bit the approach we might use in anti-doping, that we're not measuring directly the substance, but maybe some indirect indications and other markers that might indicate that somebody has used doping. And this is called what is called indirect doping detection, and what the passport, the biological passport, relies on. Only in the passport, we're not using cameras and we're not using urine samples anymore, which was the direct detection, but maybe we're using, or we should use, blood markers, hormones, metabolites to indirectly get indication of whether somebody has doped. Now, the passport is exactly this. It's an electronic record of all biological markers, or certain biological markers, that have been obtained from an athlete's anti-doping control. That can be in urine, that can be in blood. And the key issue is that for each athlete, individual reference ranges are being calculated. So a certain range within these where these markers can actually fluctuate. If an athlete is beyond these reference ranges, that means something suspicious is happening. Now, reference ranges. You all see that when you go to your doctor, he gives you the lab results and there's some numbers and you're above, like the vitamin D we've seen this morning, you need to be above 30, otherwise you're deficient. Now, how are these reference ranges defined? Many of you might not know this. It's actually quite easy. It's these famous bell-shaped curve, and I've used this one, and it's an easy one to explain this based on weight. So if we're in this room here, the average weight will be about 75 kilograms. So this is what you see on the horizontal axis. And on the vertical axis, you see the frequency. So many people will be 75 kilograms, and only very few will be 95 kilograms and 55 kilograms. So this is the variation between us. So I'm maybe 83 kilograms, Jordi is maybe 73 kilograms, so there's a variation between us, right? But now, this is only one part, because this doesn't allow you to give any information of the weight of everybody of us. We just know, oh, it's going to be between 55 and 95. Yeah, it's not very accurate. Now, of course, other than the inter-individual variation, there's also the intra-individual variation. So if myself, within this group, I step on the scale every day and I try to diet, my weight is going to change a little bit, but not much. The curve most on the right, that's somebody who tries to lose weight, you know, so he doesn't really manage, you know, so at 95 goes down to 85, so its weight undulates a little bit more. The one in the middle where the variation is a little bit smaller, more happy with the weight, the variation is very small. So that's the intra-individual variation, and the passport tries to quantify these variations and to go from an inter-individual variation to the intra-individual variation for any biological marker. Now, this variability, now it gets complicated, of course, has many sources. Now, why is something changing in the body? You know that in the morning we're a little bit taller than in the evening because our spine and the vertebral discs get a bit squeezed, so there's a little bit of variation in height. And this is the same for every marker in our body that we measure, any biological marker. There's obviously an analytical variability which comes from the analysis, and there's a biological variability which has many sources. And in the passport, all these sources of variation are being quantified through certain mathematical calculations. I'm not going to go into details. This is a bit more complicated. I don't understand all of them. Now, just to give you an example how this now works. We take a certain lab value that you might check with your doctor to see your kidney function, for example, called blood urea nitrogen, but you could take any variable. And you will have a measure here, which is illustrated with the blue dot. This is the current measure that you have, around 14. Now, the doctor will tell you, yeah, fine, you're in the reference range, and the reference range are these red triangles, and these red triangles basically represent, more or less, the width of the bell-shaped curve that we just had. So that means that 99% of the population will have a value within this. But this is not great information, because obviously the reference range is huge. So you want to have some information. Okay, you're a woman, maybe your value is really low, and how can you factor this in to make it a bit more accurate? So this information that you're a woman and that the first value is relatively low, mathematically can put this in and adapt the reference range for predicting the value of the second sample. Now, if you do that continuously with the second value and with more values, your individual limits will adapt iteratively. So coming from a very wide reference range and a definition of abnormality, you come to a very narrow reference range and definition of abnormality. Needless to say, if something goes above these limits on the right side, you will notice abnormality much earlier than if something goes on the limits on the left side. Now, this is what it looks like in real life. You see here the blue values tracked by from an athlete, hemoglobin values, and then an upper limit and a lower limit. And if the athlete is above these limits, then something is wrong. This is a real profile. Obviously, there's not only one marker, but there's a couple of markers that are also calculated. And then the interplay between the markers also plays a role for the evaluation. And the process is this, that if you have these markers in the database, the profile is generated, then experts look at these profiles and determine likelihoods of what these profile represents. Is it normal variation? Does it maybe represent a pathology, a disease? Or maybe doping? Or they're not sure and they say, well, this athlete must be targeted. Now, then this recommendation is given to the stakeholders, as many anti-doping organizations have introduced the passport now. And this brings us to the efficiency of the passport. Now, this costs a lot of money, and it's always about money, how to pay for these tests, and does it really work, and all these things. Olivier has already alluded to that. And just to give you an idea, since 2010, there's about 50 cases that have been opened on the passport, but that's not all. From targeted tests based on passport information, the number of positive EPO tests has gone up by 240%. So that means, based on the information on the variation here, you can see, oh, something is wrong, but you don't really know what. But this athlete is suspicious, so test them a little bit more. Then you're very likely to get a positive test with the conventional methods. There's also another effect that is a little bit more difficult to measure, because obviously these athletes are tested quite often. And that's a scary thing if you're doping, you know. So if you're doping and you're tested, you think, oh, maybe I shouldn't, or maybe stop because otherwise they're going to catch me. So there's a certain deterrent effect to the passport, and we can actually quantify this deterrent effect. These are abnormal reticulocyte values. Reticulocytes are young blood cells, don't worry, you know. Abnormal values, blood values, in percent, in a collective of professional cyclists from 2001 to 2010. And you see in green the values that are really high, and in pink, reddish color, the values that are really low. So either high ones or low ones are abnormal. Usually they're around 1, right? So you see that in the beginning of the years 2000, you had about between 10 and 14% of abnormal values, mostly high values. And then all of a sudden it changes. No more high values, only low values. And then all of a sudden in 2008, again, there's a big change, almost no more abnormal values. The abnormal values drop from more than 10% to about 1 or 2%. This is what you expect in the normal population. Now, this can clearly be related to efforts in anti-doping. In 2002, the EPO urine test was introduced. So what changed was just behavior of the athletes. The values were still very abnormal, but the overall values didn't change. But what made the real big impact in the abnormal values is actually the passport. You can see that 2008 onwards, this is where the passport was introduced, the number of abnormal values was heavily reduced. And that's an indication of that thing actually works. That works by deterring people of using doping, or maybe being more careful in using it, And thus, if they're a bit more careful, that's a success for anti-doping because the performance benefit is not going to be that big. Of course, they always find ways around it, and we know how they do it nowadays. They constantly adapt. So we're no fools. So this thing is actually efficient, the passport. I have explained you now how it works in anti-doping. Now, how could anti-doping now feed it back to health? How could the passport actually benefit health and the development of a healthy society? Everything we do in medicine and in science is based on reference ranges. So you go to your doctor, you have the blood test, you have the reference ranges. You measure the size of your baby and in your wife's body, you have reference ranges. But all these reference ranges are population-based. So you track a certain number over time, and if you go over population-based reference ranges, something is abnormal, and then you start other investigations. If you now apply individualized reference ranges, just as we do it in the passport, for other measures, any biological measure, it works for any measure, as long as you can quantify the variability, then you might possibly pick up diseases a little much earlier than if you use population-based references. So there, it could actually be useful if you have markers for diseases to use this approach developed for anti-doping in health, in medicine, and build something like a biological health profile. Now, some examples. If you break your arm, you play football, you break your arm, they will put you in a cast for 6 weeks. My colleagues will do that. Now, the problem is 6 weeks always enough, or is it too much for some people? For you, it might not matter, 6 weeks of work is fine, but for a professional athlete, it's an issue, 4 weeks or 6 weeks, that's at least 2 match days, that's a big point. So if you can return 2 weeks earlier, that's great. If we find now some biological markers to predict the healing of the bone, this could be an issue, we could improve this. More from a public health perspective, because we spoke about public health, for every disaster that happens, there's always a shortage of blood donors. So if there's big car accident or catastrophes, the population is usually asked to give blood. Now, usually you're only allowed to give blood every 60 days, because after 60 days, the blood you have will be recovered. That's always a problem in people with rare blood groups. There's not enough donors. Now, we know from our own research that the recovery of the blood that you gave, this one blood bag, is very individually very different, between 20 and 60 days. So the period they put in by law, you have to wait 60 days before you give blood. That's for sure. That's the sure side of things. But there's actually people who could actually give blood after 20 days. So that would save a lot of resources and make the whole blood donation process much more efficient. These are just 2 examples of how this approach from anti-doping could benefit health. Sports medicine in the first one, or traumatology, and maybe public health in the second one. I'm going to go a bit more deeper into this and develop this a little bit more for the future. Now, what we're doing at the moment is we're looking at the phenotype. We're looking at how tall somebody is, what the blood values are, the hemoglobin and all these things. This is pretty direct. But of course, below the phenotype, you all know that, and it's very fancy at the moment, there's different other levels. There's the genetic level, there's the proteomic level, there's the metabolomic level. I'm not a fair believer in all the genetic stuff because it's way too complex, and I think we're not there yet. So we have to scratch the surface a little bit and look at the level that is just beneath the surface, and that's the metabolomic level. The metabolome is basically all the products that your proteins in your body produce, the food stuff and all these things that comes out of your enzymes. And there's about 15 to 20,000 of those. And you can analyze them, for example, in the ADLQ research lab, pretty easily using certain methods. And it gives you something like this, like a metabolomic profile. Now, if you do prospective studies, and there are many of those studies, there's the nurses studies, the Framingham study was probably the first one some of you might have heard, where a certain group of population is monitored over many years and checked regarding the discovery and the development of certain diseases. And if you take the samples all along, analyze them with this, and then with the passport mechanism and the combination of several markers, you'll be able to identify certain metabolomic patterns through this passport approach and identify certain candidate metabolites that will help you in identify these diseases or certain diseases in the population a little bit earlier or much earlier. An example how this works, and I'm very grateful to my friend and colleague Pierre-Edouard Sotas, who has developed this and who has had the courtesy to provide this brand new information to me, so it's not my merit, it's his. Prostate cancer. You know that prostate cancer, big health issue, all the men can go for screening after 50 years. If you had a brother or a father affected, you can go younger. There's 80 million tests a year. And a lot of tests, 12 million of those, come back positive. If you have a positive prostate PSA test, prostate-specific antigen, that's the blood test, then you have to go for follow-up investigations. And that's very expensive. Some of them are invasive, so they do biopsies in the prostate. And of these biopsies, 75% come back negative. And this is because there's another condition called benign hypertrophy of the prostate that shows also high PSA values. Of course, you will pick up all the cancers, as you can see here on the left panel, but you will always catch all the hypertrophies which are not bad. So the test has a very high sensitivity, the ability to find the true negatives, but has a very low specificity and ability to find the true positives. So that's a problem, and you can imagine how much money there is behind this. Now, if you find something, a metabolomic approach, to do this, to improve this, this would be a huge leap for healthcare. These are the results on a metabolomic profile for prostate cancer. You can see that both the metabolomics and the PSA, they have a quite high sensitivity. So the negatives we detect quite easily, but then to differentiate between the hypertrophy and the true cancer is actually much, much higher in the metabolomic approach than in the PSA approach. This is just one example. There's other examples for ovarian cancers and for melanoma. And I want to summarize this as a little outlook that in this 10-15 minutes you've learned what the biologic passport, how it works, that it's a longitudinal monitoring tool for any biological marker in the detection of doping. Statistical modeling is used for that. It can be used as a sanctioning tool, as a targeting tool in anti-doping, but it can also be applied to any biological measure and has therefore a huge potential for the detection of pathologies. Thank you very much.
Thank you very much, Olaf. Very interesting presentation.